University, Faisalabad. I would like to discuss pH, buffers, acid-base balance. Acids. Acids are chemicals which dissolve H plus ions when dissolved in water, HCl. Strength of an acid. It is the degree of an acid to dissociate or release H plus ions when dissolved in water. Strong acid dissociate completely, release all of its all of their H plus ions. Weak acid do not dissolve completely, release only some of their H plus ions. Human body produces two types of acids, volatile and non-volatile. Volatile acids. Example is carbon dioxide. Carbon dioxide itself is not an acid, but it forms carbonic acid, a weak acid, by combining with H2O. CO2 plus H2O is equal to H2CO3. It is produced during the aerobic metabolism of cells. H2CO3 dissociate into H plus and HCO3 bicarbonate. H2CO3 plus H2O is equal to HCO3. Formation and degradation of H2CO3 is reversibly catalyzed by carbonic anhydrase, non-volatile acids, also called fixed acid. Examples are HCl, sulfuric acid, products of protein catabolism, phosphoric acid, product of phospholipid catabolism. Other fixed acids which are produced during disease process are ingested are uh, keto acid, lactic acid, and salicylic acid. Chief acids of the body, carbonic acid, which is formed by the reactions between water and carbon dioxide. H2O plus CO3 is equal to H2CO3. Carbon dioxide is considered as an acid component which is volatile. Organic acid, for example, lactic acid, formic acid, citric acid, malic acid, oxaloacetic acid, mineral acids, phosphoric acid, and sulfuric acid. Alkalis. Alkalis are the chemicals which dissociate OH or hydroxyl ions when dissolved in water, QOH and NaOH. Bases. Bases are the chemicals which conjugates with H plus ions, except H plus ions. For example, HCl is an acid which gives its H plus ions when dissolved in water, and chloride is known as the base of HCl, with which H plus ions is conjugated. Chief bases of the body: bicarbonate, sodium and potassium salt of oxalate and citrate from NaHCO3 and KHCO3 respectively. Hemoglobin, for example, in RBC, oxyhemoglobin in RBC, proteins, phosphate, monohydrogen phosphate, anion of organic acid, pyruvate, lactate, and citrate. Acid and their bases. Strong acids have weak bases. Strong acid, when dissolved in water, release all of their H plus ions, dissociate completely. This is because the bases of these acids are weak and thus cannot hold their H plus ions strongly. Weak acids always have strong bases. Weak acids, when dissolved in water, do not release all of their H plus ions, do not dissociate completely. This is because the bases of these acids are strong and thus hold their H plus ions strongly. pH. It is the negative log of hydrogens. pH 7 is neutral. pH greater than 7 is alkaline. pH less than 7 is acidic. Increased pH of blood causes alkalosis. Decreased pH of blood causes acidosis. How to detect acidity and uh, alkaline? Acidity and alkalinity can be detected by the following methods. Litmus paper, it changes its own color when exposed to acidic or alkaline solution. It turns red with acid and blue with alkaline solutions. Chemical indicator, they detect the range of pH of a solution. Congo red, phenolphthaleins and methylene blue. pH meter, it determines the exact pH of the solution. Water dissociation, it is the decompositions of the water into its component ions, H plus and OH ions. H2O is equal to H plus OH. The ionic value of H2O is 10 minus 14. Therefore, in pure water, H plus ions is equal to HOH plus OH minus is equal to 10 power minus 7 gram mole per liter, which means the pH of water is 7 neutral. Henderson Hasselbalch equation. Derivations of HHB equation is not important to remember. The equation is pH is equal to pKa plus log salt over salt. pKa dissociation constant, which is known and fixed for every acid. Importance of HHB equations. If the pH of a buffer solution is known, then the ratio of salt over acid can be calculated and vice versa by HHB equation. For reversible reactions, the buffer solution of known pH can be prepared by adjusting the ratio of salt over acid. By calculating salt over acid ratio, one can also assess the absorbable amount of medicine at a given pH. Maintenance of blood pH. The pH of blood is 7.4. The pH is maintained by buffer system, bicarbonate buffer system. HCO3 over H2CO3 is the chief buffer of blood. The ratio of HCO3 over H2CO3 is 20 ratio 1. pH is equal to pKa plus log or salt over acid. pKa of H2CO3 is 6.1. Salt is HCO3. Acid is H2CO3. Ratio of salt over acid is 20 ratio 1. Therefore, pH is equal to 6.1 plus log 20 over liter 20 over 1. pH is equal to 6.1 plus log 20 minus log 1. pH is equal to 6.1 plus 1.3 minus negligible. pH is equal to 7.4. Maintenance of normal pH. Following are the natural system which regulates and maintain the normal pH and also provide defense against changes in the normal pH. Acid-base buffer system, respiratory system, urinary or renal system. Acid-base buffer system. Buffer solution. It is a solution which contains a weak acid and its salt, sodium or potassium salt, or a weak base and its salt, sodium or potassium salt, and it resists the change in the pH when a small amount of the an acid or alkali is added to it or removed from it. Chief buffer system of the bodies are bicarbonate system, HCO3 over H2CO3, phosphate system, HPO4 over H2PO4, protein buffer, protein over H proteins, hemoglobin buffer, HB over HB. Following are the buffer present in various body fluids. Plasma, main buffer is bicarbonate, HCO3 over H2CO3, and to a lesser extent, protein, H proteins. Extracellular fluid, main buffer is bicarbonate, HCO3 over H2CO3, and minor buffer is phosphate, HPO4 over H2PO4. Phosphate is the most important urinary buffer. Intracellular fluid, main buffer are proteins, protein over H proteins, and phosphate, HPO4 over H2PO4. RBC, main buffer is hemoglobin, HB over HHB. Mechanism of action of bicarbonate, and NaHCO3 over H2CO3. When a strong acid is added to a buffer solution, it reacts with the salt, sodium or potassium of the buffer and is converted to a weak acid, HCl. Strong acid, HCl plus NaHCO3, NaCl plus H2CO3. When an alkali is added to a buffer solution, it reacts with the acid of the buffer and is converted to weak salt, NaOH, alkali. NaOH plus H2CO3 is equal to H2O plus NaHCO3, NaHO3. Buffering action against volatile acids, CO2. CO2 is the main volatile acid produced in the tissues. The main buffer for carbon dioxide is Hb over HHb. Carbon dioxide enters the bloodstream, reacts with H2O, and forms carbonic acid by the actions of carbonic anhydrase, CO2 plus H2O, H2CO3. H2CO3 is decomposed to H plus and HCO3. H2CO3 is equal to H plus and HCO3. HCO3 enters the plasma and is replaced by chloride to maintain electrical neutrality. H plus ions are buffered by deoxyhemoglobin. This buffering prevents change in the blood pH. H plus plus uh, HB is equal to HB. Deoxy HB is much more efficient than oxyhemoglobin in buffering against carbon dioxide in the lungs. Reverse reactions take place and carbon dioxide is eliminated. Buffering actions against non-volatile acids are fixed acids. The most important and the first line of defense against fixed oil is HCO3 acid is HCO3 over I2CO3 buffer system. HCl is the main fixed acid and it is buffer in the following way: HCl plus NaHCl CO3 is equal to NaCl plus I2CO3. NaOH plus I2CO3 is equal to I2O plus NaHCO3. As shown above, hydrochloric acid is a strong acid is converted to by bicarbonate to carbonic acid, which is a weak acid. This H2CO3 is then further decomposed to I2O and CO2. CO2 is finally eliminated through respiration. Respiratory system. In response to any disturbance in blood pH, mainly due to carbon dioxide, lungs alter rate and depth of breathing in order to keep the pH within normal limits. In blood, increased carbon dioxide causes acidosis, less pH, and decreased carbon dioxide causes alkalosis, increased pH. In case of acidosis, lungs increase the rate and depth of respiration, eliminating excess of carbon dioxide. A reduced level of carbon dioxide will result in decreased formation of bicarbonate. The result will be increased HCO3 over H2CO3 ratio, and the pH of blood will increase back to normal. In case of alkalosis, lungs decrease the rate and depth of respiration, resulting in retention of carbon dioxide. This carbon dioxide is then converted to I2CO3. The result will be decreased HCO3 over I2CO3 ratio, and the pH of blood will decrease back to normal. Urinary or renal system. Kidney maintain blood pH within normal limits by controlling the reabsorption of HCO3 into blood plasma and excretion of H plus ions in urine. Reabsorption of HCO3 bicarbonate. Normal glomeruli reabsorbs almost 100% of HCO3 and bicarbonate reabsorbs only 10%. Assalamu alaikum. I Dr. Mohammad Akram, Department of Eastern Medicine, Government College University, Faisalabad.